Okay, we could still chat to wait. So how's the weather, I mean, in Boston?
I think it's been similarly weird. Warm some days, wet, snowy. I think if it hadn't been so warm, we would have several feet of snow this year, but not any at all so far.
Okay.
So.
Wow. Yeah. I kind of feel the climate crisis is near. And you know what? I actually visited Boston the other day, in November actually, to give a bunch of different talk to, different university at that time, also attending the conference. Oh my God, in November in Boston, I literally kind of walked from Boston University to Harvard University after my talk at the Boston University, and I don't want to just take the T or whatever. The metro system is crazy. Over there, the Green Line especially. But I just literally walk because the weather is so beautiful. Out there, the sunny and then very warm. And then I just decided to just walk. 30 minutes, and that's the best strolling. That's last year. I ever had. So very weird.
Yeah, Boston's a relatively small city, as you know. You can get pretty far by walking in a fairly short amount of time.
But when I was student and postdoc over there, I never imagined kind of walking from one place to the other. I always kind of use my car. But oh my God, that day, I cannot even imagine just taking the subway because weather was so beautiful. Entire the week, basically the weather was so great. In November in Boston, yeah, I never imagined.
It's unusual.
I heard the area, the Colorado, because of fire and then snow afterward, the snow basically helped extinguishing whatever fire problem at the time, right?
And we just got a couple inches last night, but it gets so warm in Denver periodically that it doesn't build up much in the city, but we're getting loads of snow up in the mountains. So it's actually kind of nice. You don't have to shovel as much, but then a 2-hour drive, you're skiing in a couple feet of snow.
Okay. And then this is your house right now? This is beautiful. You have very nice, flowers.
They're fake flowers. This is the basement, but
a basement. Your basement is much better than my, the living room actually.
Well,
okay, so go ahead.
Just they're dried flowers, I guess. I shouldn't cut them short. They were a gift from a friend.
Okay, great. All right, I guess we probably want to start. So, okay. So, good morning, afternoon, evening. Hola, hello, bonjour, ni hao, konnichiwa, annyeonghaseyo. Hello all. Welcome and thank you for joining us, the 15th seminar and Nature Chemical Biology Day 2 and the first SynBYSS meeting in 2022. I was so excited. And of course, Happy New Year to everybody. And I hope you have enjoyed holidays and you are still keeping your New Year resolution. I have enjoyed unbelievably warm St. Louis winter break near 60 and up to 70 Fahrenheit for a week or longer, although we will have single digit today with snow covered road already. I guess the first real snow this winter and a little bit, of course, unusual and freezing right now, or climate crisis. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Dr. Terry Sheppard. I would not need any introduction of him, but he's the editor-in-chief of Nature Chemical Biology. Also, he's the second pioneer who joins us from the same journal after Dr. Caitlin Dean last September. So I'm so grateful for Nature Chem Bio support for this seminar series. Also, Dr. Terry Sheppard, basically said he will stay even after this 1 hour session for the separate Zoom. So if you want to ask any question about journal editing or publishing, please join us for another Zoom meeting. So Terry, the virtual floor is all yours now. Thank you.
Thank you very much, Taesuk. It's a real pleasure to be with everyone, and I really appreciate your efforts in organizing this virtual seminar series. I think in these strange times, it's really critical that we continue the scientific conversation, and these types of forums are really, really useful in doing that. And I also really like the format that you've chosen because it gives us more senior people very little time, and it places the emphasis on the most exciting research that's going on in the community in synthetic biology. So I think that's a great way to do it. And finally, I've organized things before, and I know how much time and effort this takes. So thank you, and we all really appreciate what you're doing here. So thank you. So I put together a few comments that are a bit historical in perspective, I guess, on the field. I've been the chief editor of Nature Chemical Biology since it started in 2005. And before that, I was a chemical biologist myself. I trained as a bioorganic chemist, and then I did a postdoc more in the molecular biology space, and had an independent program focused on nucleic acid chemical biology. And at that time, in the early 2000s, there weren't many forums to publish interdisciplinary research. There was really only 1 or 2 chemical biology journals to speak of. And so when I heard that Nature was interested in doing such a journal, I really jumped at the chance, and I feel I've found my calling in some regards. And I think what's been most exciting about it, about this opportunity, is that it allowed us to develop a journal for a field that is developing and evolving in real time. And it's very exciting. And the challenge to identify papers that really reflect the diversity of chemical biology research is really exciting and humbling in most respects. The job of the editor is actually quite an exciting one, because every day you see something new. You're always learning. You find out about that new technique that's coming down the pipe, and also the cool new hypotheses that are governing different facets of the field. And I think the greatest part of the job for me is working and engaging with a really diverse group of scientists from around the world who make these discoveries and make it all happen in communicating their results to the community. So it's really been an exciting time, and we celebrate our 17th anniversary this next summer, so we're looking forward to that. So having been doing this for a while, I've been witness to the evolution of chemical biology and synthetic biology really in parallel during my career. And I've always considered these two fields to be kindred spirits, because they approach problems in very similar ways, and their way of thinking about the solutions is quite similar. And so I just wanted to make a few comments about that. Chemical biology, which I would say really came together in the 1990s, started as a field that was very much focused on small molecules and chemical tools that were designed to understand biological systems, right? And then since then, it has really expanded to include a lot of different chemical biologists, or chemists and biologists and engineers of various stripes. And they've taken a much broader view of both the research problems that they're focused on, but also in bringing together the tools and thinking of different disciplines: chemistry, biology, engineering, computational science, mathematics. And for a chemical biologist, the main motivation is often what can I do to understand mechanism in biology better, right? That's one of the fundamental principles that chemical biologists bring to the table. But from my point of view, chemical biologists have always had a restless streak. That is, it's not good enough to just understand how biology works. You want to use that knowledge to make new things, build new non-natural systems that have practical applications or medical applications. And that's really been a thread that started in chemical biology way back since bioorganic chemistry in the 80s and 90s. And it's this particular thread, I think, where synthetic biology and chemical biology really overlap. And if you think about the Venn diagram between those two fields, if they were at first just touching, now they're starting to really overlap in some key ways. And I think about fields like metabolic engineering, right, which we'll hear some about today, small molecule tools, protein engineering, biosensing, expanding the genetic code, regulatory networks and gene circuits, and CRISPR, of course, these days. Designing synthetic signaling systems that could be useful in things like CAR-T systems and so on. And all of these problems are things that both chemists, chemical biologists and synthetic biologists work on. And I really do believe that working together more in the future on these problems will really accelerate discovery in these fields and also the practical applications that they promise to science and to society. And then finally, I think there's a lot that the two fields can learn from each other, even though they've kind of pursued parallel tracks in many areas over the years. I think that the chemical perspective that chemical biologists bring to the table and the emphasis that they put on molecular understanding and mechanism and characterization of systems at the molecular level, I think is really useful when synthetic biologists think about design and iterating systems to make them better or more efficient or more practically useful in cells or organisms. Synthetic biology, in turn, I think is really a very practical engineering perspective on things, that there's a defined goal and we set out to achieve that goal through the technology and the approach that we use. And I think that practicality is a real value of the synthetic biology community. And the other thing I've always admired about synthetic biology is the openness of the culture. They've always had this open science perspective where data and reagents are shared, and various conferences that everyone is aware of to present their new parts and pieces. I think it's fantastic. And this is a thing that broader science could learn from more and more as the years go on, is sharing data and facilitating the conversation to advance knowledge and practical insights down the road. So even after all these years, I feel even more excited about chemical biology and synthetic biology and the potential that these fields bring to science generally, but also the things that are going to come in the future as far as applications in medicine and addressing things like the climate crisis and so on. So from my seat as an editor, I'm just especially looking forward to witnessing this evolution and interacting with all the creative scientists and seeing what results. So that's all I have to say today. Thank you very much.
Oh my God, wow. Thank you so much for your amazing talk and inspiring insight with the history. You know, I was trained as an applied chemist during my BS program, but I am doing synthetic biology right now. And then I didn't take the, advanced kind of biology course when I was a high school student. So I completely agree with you regarding interdisciplinary research and its importance. And also, I also believe I met you for the first time at one of the synthetic biology Golden conferences, I believe. So, of course, I guess the joy of being editors is reading exciting papers, but the worst part is to reject papers, right?
Absolutely. I mean, there's so much good work that comes in the door, right? And we, Nature journals at least only publish about 10% of what are submitted, and about a third of what gets submitted gets reviewed. So it's a very, we spend a lot of time rejecting good papers, and it's difficult. It's really difficult.
Sure. Now we'll hear, I mean, more after this session. I mean, during the separate session, I mean, we want to kind of ask more questions. Okay, so now the main speaker of today with a longer introduction, and Allison Werner is a metabolic engineer and biologist that scientist in the Renewable Resources and Enabling Sciences Center at the National Renewable Energy Laboratory. Ellie was a postdoctoral researcher in Greg Beckham's group at NREL from July 2018 to June 2021, before she joined as a staff member. During her postdoctoral studies, Ellie worked on many projects aimed at developing microbial biocatalysts for the valorization of lignin or plastic feedstock, resulting in 7 publications. And that's why I love her work, because of my passion is also toward plastic upcycling and valorization of lignin. And of course, I mean, Ellie earned her PhD in cell and molecular biology from Colorado State University in May 2018 under Christy Peebles. I know her very well as well, since she was, I think, at MIT. Her dissertation work developed synthetic biology tools and elucidated system-level metabolomic understanding of cyanobacteria in the industrially relevant condition of light dark cycles. Ellie earned her Bachelor of Science in Biomolecular Engineering in May 2014 from the Milwaukee School of Engineering. So without further ado, Ellie, take it away. It's all yours now.
Thank you. Let me get my slides up. Are you seeing, what are you seeing right now? The full screen? Yes, can you hear me?
Yes.
Okay, great. Sorry about that. So thank you so much for the kind invitation, Dr. Moon, Professor Moon, and thank you all for your time today, and Happy New Year. So today I'll be presenting some of, oh, sorry, something weird just happened. Are you still seeing the full screen?
Yes.
Sorry. Okay, great. So today I'll be presenting some of the work that Professor Moon just mentioned, using Pseudomonas putida as a biocatalyst for the valorization of lignin or upcycling of plastics. And it'll primarily really be about plastics today with 2 deep dives. So I realized when putting this seminar together that many of you may not have heard of the National Renewable Energy Lab. I think you can see my pointer, yeah. So we are located in Golden, Colorado. This is our main campus. We also have a wind campus. So both of these are really at the foothills of the Rockies, as you can kind of see in the beautiful backdrop we have. To place us within the National Lab System, I pulled this graphic from the 2020 State of the DOE National Laboratories report. And you can see all the labs are arranged on a scale from science to technology. NREL sits just right of center as an energy technologies lab with the stated mission to advance the science and engineering of energy efficiency, sustainable transportation, and renewable power technologies, and provide the knowledge to integrate and optimize energy systems. With that mission statement, it might not be a surprise that we're primarily an EERE, or Energy Efficiency and Renewable Energy Lab, but we also have funding from the Office of Science, among many others. So we're a relatively small lab with just under 2,300 full-time employees. We also host postdoctoral researchers, as I was, and undergraduate and graduate students through joint appointments with other universities. I sit within the Bioenergy Science and Technology group, shown here is a pie chart of our business volume last year. And so we're a decent sized chunk with quite diverse research, all unified by the goal to bring biofuels and bioproducts to market. My work specifically aims to develop technologies for the valorization of lignin and plastics, the two topics for today. But before I dive in, I want to give a brief introduction to lignin. So lignin is the second most abundant biopolymer on Earth, second only to cellulose. This is a depiction of an example structure, and you can see it's this complex organic phenolic biopolymer. It has different units, it also has different inter-unit linkages, so it's very chemically complex, which in nature, it provides rigidity to woody plants and trees. In large-scale industrial bioprocesses or processes converting cellulosic biomass into fuels and chemicals, lignin is left behind and underutilized, often just burned for heat. But as you might imagine from looking at this structure, there's a lot of interesting and high value products that can be made from lignin, including plastics, carbon fiber, and fuels. So given the carbon and economic motivation to valorize lignin, heterogeneity is a major barrier towards the upgrading. So the polymer deconstructed, be it by thermal, enzymatic, or catalytic means, results in this really heterogeneous slate of chemicals. And so you might see some of these and think, oh, those are very valuable. The problem is separations, and this can be a pretty challenging and costly endeavor. So an alternative strategy to using classical separations is using a biocatalyst, and this is where the term biological funneling comes in. And so what we mean by funneling is the conversion of these diverse chemicals into a single central intermediate. And so many bacteria contain native pathways for the utilization of these lignin-related compounds, so-called upper pathways. So then we can engineer conversion of these central intermediates into products of value, like what's shown here is Beta-Keta Adipate, which is a precursor for performance advantage material. Alternatively, in nature, these compounds are catabolized, generating cell growth or biomass via the TCA cycle. Pseudomonas putida, and in our case we use the strain KT2440, has become a widely used chassis for biological funneling of lignin-related monomers, such as those shown here, for a whole host of reasons, but notably tolerance to stress, so it can survive and even thrive in harsh environments that kill other microbes. The ability to accumulate biopolymers, specifically polyhydroxyalkanates or PHAs, and the availability of genetic tools for modification of the organism relatively easily. So here I'll quickly run through some recent advances in the lignin space with this hopefully not too silly cartoon. So this is supposed to represent a Pseudomonas putida cell with the lignin polymer and the monomers here. So one of the first efforts I was involved with after joining NREL was searching for aromatic transporters. So basically asking the question, how do these compounds enter the cell? And it's thought that much of this occurs passively, but in specific strains that we have engineered in specific bioprocesses, uptake is actually a bottleneck. So we wanted to identify these transporters in order to engineer improved uptake. In collaboration with Adam Feist's group at DTU and UCSD, we used adaptive laboratory evolution to identify transporters for coumaric acid and ferulic acid. And this led to a collaboration with AJ Masai's group, where we verified both an MFS symporter for coumarate and ferrolate, as well as ones for vanillate, protocatechylate, and 4-hydroxybenzoate. Moving to another project, as I alluded to a couple slides ago, there are several different link units in lignin, and there's 3 canonical types, S, H, and G. Hopefully you guys can all see my pointer. And so to maximally convert lignin towards economic viability, we want to access all 3. So by employing a suite of tools, including transcriptomics, proteomics, biochemistry, and metabolic engineering, we elucidated the native pathway for catabolism of syringate, the compound shown here, in Pseudomonas putida. And then we built a strain that can convert all 3 canonical types to a single product, shown here as 2-pyrone 4,6-dicarboxylic acid, or PDC, which is a promising precursor for biopolymers. In a similar effort, similar meaning we're expanding the so-called biological funnel to access more of the lignin, we engineered utilization of a dimer. So in the deconstruction methods we use to convert lignin to break down lignin, many of the methods are pretty good at cleaving CO bonds, but not as good at cleaving CC bonds. So in collaboration with Josh Michener's group at Oak Ridge National Lab, we used TN-seq and Novostingobium aromaticivorans to identify a gene potentially responsible for the conversion of this beta-1 linked dimer into a lignostilbene compound, which can be cleaved into 2 units of vanillin via dioxygenase. We then engineered the strain into Pseudomonas putida and demonstrated conversion of 1 unit of dimer into 2 units of muconic acid, which again is a precursor to advanced materials. More recently, we've improved the rate of muconic acid formation, so this product here, from coumarate by debottlenecking the pathway, meaning making, well, in this case, we specifically were debottlenecking 4-hydroxybenzoate hydroxylation. And then lastly, not necessarily biological funneling, but a really fun project that I wanted to mention is gram-negative bacteria studied to date, all of them studied to date, secrete these outer membrane vesicles. So this is supposed to show the blebbing event, and blebbing is the scientific term here, for the secretion of vesicles. And we found that Pseudomonas putida, grown in lignin-rich media, not only secretes vesicles, but that they are enriched with aromatic catabolic enzymes, and those enzymes retain their activity, which is really cool. We don't understand the physiological role, but we are working to do that. And we're also interested in these vesicles as a synthetic biology tool. So putting this all together, our group takes an integrated approach to lignin valorization, beginning with lignin deconstruction, which generates this heterogeneous chemical mixture that is microbially converted into a single product, which then can be separated from the fermentation broth and polymerized into new materials, often with advantages over the petrol-based analogs. So kind of drawing the parallel to plastics here, as our group has more recently begun to address plastics, fundamentally, both biomass and plastics are low value, dispersed, heterogeneous solid polymers. So if you look at biomass, biomass is extremely complex and has more than 3 components, but the 3 major components being hemicellulose, cellulose, and lignin. Now, if you look at a plastic bottle, you have a cap of polypropylene often, a label of polyethylene, and the bottle composed of polyethylene terephthalate. If you think about commingled plastics, it gets even more complex when you have, polystyrene cups or polyvinyl chloride pipes, or even multi-layered packaging, which is layers of composites or many types of materials. And so this heterogeneous bit is similar between these 2 feedstocks. So given the parallels, we're attempting to leverage NREL's rich experience in biomass conversion towards the plastics problem. So thanks to Lars Blanc's fantastic introduction to the plastic crisis and the role synthetic biology can play a few months ago, I'm only going to spend 1 slide introducing the plastics problem. But an astonishing amount, 8.3 billion metric tons of plastics have been manufactured, and that's just of 2015. And to me, I didn't even know how to wrap my mind around what 8.3 billion metric tons is. And so Wired has this nice infographic. How heavy is 8.3 billion metric tons? That's 822,000 Eiffel Towers, or 80 million blue whales, or 25,000 Empire State Buildings, or 1 billion elephants. Just sort of an unfathomable amount of material that we've produced. And less than 9% of all of that has ever been recycled. So this is a really useful infographic from Guy Riddell's Science Advances paper in 2017, showing the primary production of plastics. A good amount is in use, and then this discarded fraction, which we know is intended in an ideal case would go to a landfill, but often ends up in the environment, or specifically the oceans. And so this is a pretty unideal system. And the World Economic Forum estimates that 20% of global petroleum consumption will go, 20% of all global petroleum consumption will just go to plastic production by 2050. So the need for recycling strategies and the development of non-petrol-based plastics is clear. The Bottle Consortium, which was jointly funded by the Bioenergy Technology Office and the Advanced Manufacturing Office, kicked off last year with the mission to develop robust processes to upcycle existing waste plastics in new bio-based plastics and processes that are recyclable by design. So starting with plastic wastes, we're developing different deconstruction technologies and really casting a wide net here, looking at thermal catalysis, electrocatalysis, and biocatalysis to break down these different polymers, which generates a heterogeneous mixture of products that can then be biologically funneled into to form new polymers. Often, and we're targeting performance advantaged plastics, recyclable by design polymers, or upcycled materials. And I'll highlight that this green arrow is really the focus of my work. So going back to the silly cartoon, I'll highlight two recent projects in the plastic space that I'll spend the rest of the seminar talking about. So polyethylene terephthalate, as I mentioned, is plastic bottle, the primary component of plastic bottles. And we used chemical deconstruction of PET to form bis 2-hydroxyethyl terephthalate, or BHET, and then built a strain to convert this product into Beta-Keta adipate, which is the precursor for performance advantage materials. Secondly, and we're just wrapping up this massive effort, looking at upcycling of mixed plastics. So commingled plastics are very difficult to do anything with. Even though there are mechanical recycling strategies for individual plastics, it gets very complicated when they're commingled. So we're working on a strategy, and we've worked out a strategy to convert these into a suite of intermediates that can then be biologically funneled into either Beta-Keta adipate or poly-PHAs. And so this project is in preparation for publication. So I appreciate Dr. Moon's flexibility in waiting to upload this to YouTube until we've published. From the top, I want to first acknowledge our team and call out some key players. Greg Beckham, my former postdoc advisor and now PI, Kevin Sullivan, Kelsey Ramirez, and Lucas Ellis, who are all co-first authors on the Mixed Plastics Upcycling Project. Christopher Johnson, who leads the synthetic biology team, Davinia Salvatrucha, who's heavily involved in the biological sciences and fermentation science in the lignin space, and Adam Gus, who's our close collaborator and lead of the bottle upcycling task. Okay, so jumping in. Recent work from folks in our group, including Scott Nicholson and Nick Rohr, used a supply chain modeling tool called Material Flows Through Industry to analyze US consumption, supply chain energy, and GHG emissions from common commodity polymers. And so you'll see things like polyethylene, polypropylene, PVC, PS, PET. The first project, and this is just a snapshot of their findings. I really encourage you to read their paper. It's very insightful. But in the first project, we were just looking at PET. So we developed a method using chemical depolymerization, followed by upcycling to Beta-Keta adipate, shown here in blue, which can be copolymerized with hexamethyldiamine to form this nylon analog, which actually has performance advantages over nylon 6,6, including decreased permeability to water and an increased TG. So jumping into the details, we use the well-established method of chemocatalytic glycolysis with titanium butoxide and ethylene glycol to convert polymers into bis 2-hydroxyethyl terephthalate, or BHET. To convert BHET to Beta-Keta adipate, we did several stages of metabolic engineering. First, taking BHET to terephthalic acid by utilizing the PETase and mHETase enzymes that have received a lot of attention in recent years, and then conversion of terephthalic acid to protocatechuic acid which is one of those central metabolites in Pseudomonas putida, by employing a transporter from Rhodococcus and 2 catabolic enzymes from Chemo. Then protocatechuic acid is converted to beta-Keta adipic acid by deletion of the native PCA-IJ. Downstream processing of beta-Keta adipate can then separate, acidify, extract, and esterify in order to form this performance advantage nylon. So I'm just going to spend a slide on each of those targets that we looked at. Oh, I forgot to say that in the conversion of BHET to TPA, 2 units of ethylene glycol are released. And so the first goal was to improve the utilization of ethylene glycol. We did this by leveraging 2 previously established methods, first being overexpression of the glycolate oxidase, GLC-DEF, using the non-native and constitutive strong promoter PTAC, and the second being deletion of GCLR, which is a repressor for GCL, the glycoxylate carboligase, GCL. The resulting strain utilized about 90 millimolar of ethylene glycol as the sole carbon source for growth. For TPA, we built a combinatorial library expressing either transport or catabolic genes from Rhodococcus jastii or Chemo E6. And so we found that, and I should say in both cases, the catabolic genes comprised a TPA oxygenase as well as a DCD dehydrogenase. And we found that the best combination was the transporter from Rhodococcus jastii TPA-K and the catabolic genes from Chemo. And the resulting strain again used terephthalic acid as the sole carbon source for growth. We connected BHET to TPA by expression of PET hydrolases, PETase and mHETase, described in the very well known and famous paper from Yoshida et al. In Science in 2016, wherein PETase can convert both PET polymers or BHET into an intermediate mHET, and then mHETase takes it further to terephthalic acid. And so by expression of PETase and mHETase in Pseudomonas putida, we enabled growth on BHET as the sole carbon source, shown here in purple, as well as utilization of BHET. And I wanted to show this plot, which is just BHET as the sole carbon source, which really shows this quite substantial intermediate accumulation of mHET. We actually see that this goes down substantially if you add a supplemental carbon source like glucose, suggesting it could be mitigated by an extra energy source. Okay, so to convert BHET to BKA, we deleted PCA-IJ, which is the native CoA transfers, and evaluated the strain in bioreactors. We fed glucose using a diostat, so every time the dissolved oxygen hit 70%, we spiked in, or the instruments spiked in more glucose, and then we adjusted the agitation manually. The tricky part is BHET is only very minimally soluble in water, so it was the Wild West, and we were pasteurizing the powder and then dumping it in through the head plate, shown here in this picture. So we have discrete BHET feedings shown by the X's, and by the end of our 96-hour experiment, we had added an equivalent of 31.5 grams per liter BHET. Interestingly, we never actually saw accumulation of BHET or mHET in solution, so it's being added in as a powder, which suggests that it's solubilizing at the same rate that it's being utilized. We did, however, see spikes of TPA throughout the cultivation and accumulation of Beta-Keta adipate, shown in blue. So the titer, yield, and productivity fluctuated across the cultivation, but at the 96-hour, the endpoint, we had a titer of 15.1 grams per liter, a yield of 0.76 or 76%, and a productivity of 0.16 grams per liter per hour. Lastly, we wanted to demonstrate a proof of concept for conversion of PET flakes into Beta-Keta adipate. And so we took bioxylate-oriented PET and did the glycolysis, generating this goo shown in the round bottom flask labeled number 2. And by NMR analysis, we see that there's no oligomers left. So we just have BHET or TPA. So then we added to water and filtered to sterilize and added this directly to the cultivations. And excitingly, we observed more or less the same dynamics as before. BHET, now shown in the square, is utilized, as well as mHET, which BHET can hydrolyze to mHET at low or high pHs, so likely the addition to water formed some amount of mHET. but both species were utilized and converted into BKA. So while this was really exciting, and we're currently further optimizing this system, we're also asking the question, what about other plastics? Or potentially a more relevant question to post-consumer streams, what about mixed plastics? So going back to the consumption, supply chain energy, and emissions plot, we focused next on 3 of the top 5 commodity materials: polyethylene, PET, and polystyrene. So in this process concept, mixed plastics, which could eventually come from post-consumer sources, which are mixed plastics are rejected at material reclamation facilities quite often, or ocean plastics or even non-recyclable plastics could feed into this mixed waste stream. But again, here we're just looking at polyethylene in blue, polystyrene in pink, and polyethylene terephthalate in orange. Then a chemical oxidation step in a one-pot reaction can act on multiple polymer types to generate this chemical mixture of oxygenates. And we consider these oxygenates to be relatively bioprivileged, as they're relatively accessible to microorganisms like Pseudomonas petita. So in the second step, the bioconversion step, we funnel this mixture into a single product. And here we're showing 2 example products, again, Beta-Keta Adipate or PHAs, which can be used in performance advantage materials. So looking first at the chemistry, we're using auto-oxidation chemistry, which is really complex, at least to me, it's very complex. These initiation reactions generate radical species that can then react with oxygen. This was really well studied by Walter Partenheimer, who applied this chemistry to the depolymerization of several individual plastics, in which case the radical initiator performs H-atom abstraction on the CH bonds to generate alkyl radicals, which then react to form hydroperoxides. Then a cobalt and manganese catalyst system can convert these into alkoxyl radicals, ideally resulting in CC cleavage through beta scission, generating these oxidized molecular products. And notably, and I think this is important to note, is that this process, generally this auto-oxidation process rather, is used at really large scales industrially for the conversion of xylene into terephthalic acid. Just a note there. So in our case, we applied this first to polystyrene feedstocks, and we used both pristine polystyrene beads as well as post-consumer EPS foam cups. One thing to know is that we know that these, especially these post-consumer products, contain a ton of additives, things like dyes or another many things, which is a massive analytical undertaking. So we are doing a massive characterization effort, but right for now, and certainly in this presentation, the additional things present in these streams is more or less a black box. But reaction on both of these materials generates a solution here in acetic acid that's translucent, so no residual material are visible to the eye, and generates primarily benzoic acid. And so in the case of the polystyrene beads, we're getting 63 mole% carbon recovery of benzoic acid, and it's a little bit higher for the EPS foam cup at 67%. For polyethylene, we used both pristine, oops, that's a typo, polyethylene HDPE beads, as well as a post-consumer product of an HDPE milk jug cut up here into tiny squares in a scrapbooking event in the lab. So again, the post-reaction solution is in acetic acid, and you can see through to the bottom, indicating that there's no visible solids remaining. And the reaction products were a little bit harder to identify here, but are primarily comprised of dicarboxylic acids, ranging from 4 to 22 carbons in length, produced at yields of 33 to 34 carbon mole%. So the exciting thing, that was sort of known before, but without the commodity products. So we found that it worked well in the commodity products, and then the exciting part was looking at actual mixed plastics. So we applied this chemistry to mixtures of either beads of polyethylene and polystyrene, or polyethylene milk jug plus polystyrene cup. And we found that we got retained similar yields in the mixed case. And here I'm showing a pie chart of the substrates as prepared for bioconversion. And you can see the lion's share, maybe just more than 0.5%, 50% is benzoate. And then dicarboxylic acids are really stacked towards the shorter chain, with the majority of the carbon being tied up in succinate, glutarate, and adipate, with a trailing off from there. When terephthalic acid, or when PET is added to the mix, we additionally get the product of terephthalic acid, with the distribution of DCAs remaining about the same. So my goal was then to build a strain that can convert or funnel all of these substrates into a single product. Luckily, benzoate is a native substrate for growth and is actually a really good carbon source for Pseudomonas petita, even though it's a preservative for against other bacteria. So we get conversion of benzoic acid to catechol, and then orthocleavage further down the Beta-Keta adipate pathway, going through Beta-Keta adipate itself, and finally to a unit of acetyl-CoA and succinyl-CoA. Terephthalic acid utilization, as I just talked about, was conveniently tackled earlier, and so we leveraged the strain engineering that I just discussed in this strain as well. The goal then was to look at these aliphatic compounds. So Pha Petita KT2440, these are some growth plats, grows well on succinate, C4, and glutarate C5, but does not grow on C6 through C10 DCAs. Acetate or acetic acid is converted into a unit of acetyl-CoA, which feeds into the TCA cycle. Succinate additionally feeds into the TCA cycle. And there's two reported pathways for glutarate in Pha Petita, either hydroxylation through L2 hydroxyglutarate or the well-studied gluteryl-CoA dehydrogenation pathway. So then we looked towards C6 plus DCAs. Several works in this space were really key towards this goal. Firstly, Park, Garcia, and Ornstein identified a pathway for beta oxidation of straight chain DCAs in Acinetobacter bailey, and really described the pathway in great detail in 2001. And indeed, our growth assays with ADP1 show good growth on C4, 6, 7, 8, 9, and 10, but interestingly not C5. Yannick Ackerman, Lars Blank, and Nick Works and their colleagues adeptly applied the ADP1 pathway in Pseudomonas petita for the utilization of adipate and published this work in last year. And additionally, we found some insight from a paper led by Mitchell Thompson and their colleagues where they used T and seq to identify genes essential to the utilization of fatty acids, which we consider to potentially be relevant towards metabolic pathways for dicarboxylic acids. And I had to shout out this 1970 article in the European Journal of Biochemistry from Hohensteiner, which has some really interesting insights into higher order DCA utilization in a pseudomonad, though of course, no specific genes were identified, and this is a different species. So putting this all together, we utilized the DCAP porin, the DCAK symporter, to import at least adipate into the cytoplasm. The DCA-IJ acyl-CoA transferase generates the adipoL-CoA ester. And then the DCA-A acyl-CoA dehydrogenase generates trans-2,3 dehydroadipoL-CoA. This then butts up with the phenylacetate pathway, which takes over from there, the native one in Pseudomonas petita, with conversion to enol-CoA by an enol-CoA hydratase PAAF, or potentially FAD-B, followed by action of PAAH, the hydroxyl-CoA dehydrogenase, and lastly PAAJ thiolase, which generates a unit of acetyl-CoA and succinyl-CoA. We additionally deleted both PAAX, shown here and here, which is shown to repress the phenylacetate pathway in E. coli, and there was a homolog in KT2440, and then also deleted PSRA, which is shown to be a repressor of FAD-B, among others, and was shown to improve the utilization of higher chain fatty acids in the Thompson et al. Study, as well as C6 in the Ackerman study. Okay, so putting this all together, we explored a few different strategies for expression, sort of pulling out the catabolic genes from the transporters, but actually found that the ideal system here now is AW162 grown in a mixture of C4 through C14 DCAs. And we found that there was utilization of all DCAs in this strain. And so Zooming in a little bit, we actually see two sort of phases of utilization. First, C4, 5, 8, and 10, followed by the rest. So the last modification to note is similar to the PET story. By deletion of PCA-IJ here, we targeted the conversion of benzoate and terephthalate to beta-Keta adipate, while all the aliphatic substrates would still support growth. Without the deletion of PCA-IJ, all carbon sources will funnel into the TCA cycle, which it's been well studied and described that under nitrogen limiting conditions, Beta-Keta will produce PHAs. So in sum, we built two strains, AW162, which funnels everything through the TCA cycle for either growth or PHA production, and AW307, which uses the aliphatic substrates for growth and the aromatic substrates to make BKA. So next we aim to test these strains out in actual catalyzed polymers. To prepare the substrates for growth, I'm showing here both the beads and the cup. We get this crude product, which is a brownish gold, add it to water and pH adjust with sodium hydroxide, which actually precipitates out the middle catalysts that we can recover with the crude filtration step. And then we did a final filtration step to sterilize. And we add that solution directly to the cultivations. And our goal was initially to see if the strains could just grow in the substrates. And secondarily, if all of the substrates were utilized simultaneously. And I'm showing first just this plot, which is really busy. I'm going to split it out into 3 plots, but just showing it to emphasize that there's really different concentrations of all the substrates. And so in these plots, please note that the y-axis is changing. But this is the same data broken out into different time course plots. That's all occurring simultaneously. We get utilization of benzoate, shown in pink, and utilization of C4 through C10 DCAs, as well as C11 through C17. And so by the end of the experiment, the 36-hour experiment, all were utilized to completion with the exception of C6, which was like 98% utilized or something. And so we don't think that these formation, so this increase in concentration right at the beginning, we see this routinely with these catalyzed substrates. They seem to precipitate out upon addition to the media to some extent. And so we don't actually believe these are formation events, but rather just an inaccurate, slightly inaccurate T0 measurement. So adding in, so in catalyzed mixed polyethylene, polystyrene, and polyethylene terephthalate, we get utilization of both benzoate and terephthalate simultaneously with growth on the aliphatic substrates as well. Again, we're seeing utilization of C4 through C10 and C11 through C17 with slight incomplete utilization of C6 and C13. And so this addition of PET wasn't so much of a surprise as we had previously worked with the crude catalyzed PET, but we were nonetheless excited to see co-metabolism of all of these substrates. Looking towards the post-consumer products, this is now data for catalyzed mixed polyethylene milk jug as well as a polystyrene cup. And again, we see utilization of all the substrates, but notably much less growth. This prompted us to look at Beta-Keta adipate production. So switching to the strain AW307, I'm showing it just again in a single plot to kind of show you the complexity of everything that's going on. And here's the finer detail. So we see conversion of benzoate into Beta-Keta adipate with utilization of C4 through C17 DCAs. The notable exceptions here are C6 and C7. And so this could suggest, so remember this strain has PCA-IJ deleted. This could suggest that PCA-IJ, which is the CoA transferase native to Beta-Keta, is partially active on Beta-Keta adipate, or DCA-IJ native partially active on Beta-Keta adipate, or PCA-IJ is partially active on adipate or pimolate. Nonetheless, we did get conversion to BKA, and here are some bar charts showing the yields we achieved on each substrate. So looking first at polystyrene type substrates, we compare everything to a control, which is just the known substrates there. So benzoate and acetic acid, and in the control we get 79.1% yield, molar yield. Moving to polystyrene, in the EPS cup, we more or less retain our yields with 76.5 or 78.7% yields. In the mixed PS plus PE case, we observed theoretical yields on the control, which was benzoate, a mixture of DCAs and acetate in concentration similar to the actual catalyzed polymers. And then we saw an 81% yield from catalyzed PS plus PE and an 84.8% yield from a mixed polystyrene cup plus HDPE milk jug. Lastly, from mixed benzoate, terephthalate, DCAs, and acetate, we observed 72.9% yields, which was similar in the actual catalyzed polymers as well. So I'll stop or just emphasize here that the product is really flexible, as all of the strain engineers know. And so we chose BKA as an exemplary atom efficient product, but could easily shift this to a different end product, especially if there are crosstalk reactions like our data suggests that could limit advanced bioprocesses or achieving theoretical yields. So to close out, I'll end with the demonstration of PHA production from mixed catalyzed PS plus PE. So again, PHAs are an extremely well-studied bioproduct with tons of fantastic work from many people, including those in the SynBYSS community that have done really great work, both strain and bioprocess optimization to optimize and maximize yields. We did not do that here, but we did demonstrate conversion of catalyzed PE plus PS into PHAs, as well as HDPE and polystyrene cups. And the composition was more or less the same between the two, primarily 3-hydroxydecanoic acid or HAME12, followed by 3-hydroxydecanoic acid or HAME10. So with that, I'd like to thank you all for your time and attention, acknowledge again the many wonderful people who I've worked with, point you to this paper for the first part of the talk if you're interested in more information, and hopefully we'll be submitting the second part of the talk soon as well. So please feel free to reach out to me by email if you aren't able to, if you have questions and aren't able to stick around for the Q&A.
Oh my God, fantastic talk. I especially love the mixed plastic work. That's fantastic job. So of course, this is very important work to solve climate crisis we talked about earlier today. And without solving or doing action right now, there would be no future for my teenage girl and our future generation, of course, all of us. So thank you so much. This beautiful work. So I already saw one cute question from Tianyu Lee. So good job. I have a question about MHATAs and PTAs expression. Did you try to look into how these two enzymes being secreted to cell exterior, or how BHAT import into cell interior? Interesting question.
Yeah, that's a really great question. So we used the secretion signals native to Ideonella sacchiensis in our case. So we didn't play around with those secretion signals at all. And in our case, we're not trying to depolymerize the actual PET polymer. So secretion to the extracellular space isn't absolutely essential if we assume that BHAT is entering the cell. And that's actually the model that we think is happening, is that BHAT is actually passively entering the cell, and then conversion is happening intracellularly.
Wonderful. So any other question? Actually, I have tons of questions myself, but I will ask that question another session. And Terry, do you have any question or comment?
Yeah, I did have one question. When you're thinking about the production mode metabolic engineering systems, there's a ton of engineering and optimization that needs to go into that, including dealing with things like translation burden and all that sort of stuff. And I'm curious if in the catabolic mode where you're bringing in small molecule feedstocks, how much of that comes into play when you're optimizing your system? I mean, the way it looks like you were able to plug and play a little bit with some of the known pathways, which is fantastic. I'm just curious, what level of optimization do you need for something like this?
Yeah, So we haven't done, so I should say that we played around a lot with different levels of transcription, especially with the DCA-P and K, these transmembrane proteins that we know at really high levels can actually be pretty toxic to the cell. And that's actually true for TPA-K, which isn't probably the same level of burden, the type of burden that you're talking about, but actual just stacking the membrane full of these proteins we found can be pretty detrimental to the cell health. And for TPA, during the course of our evolution experiments, we actually saw the strain increase expression of TPA-K. So it actually wanted fitness advantage by having more of that transporter, which was really surprising. And so we tried increasing expression of the DCA-K and P as well for adipate import or other DCAs, and found that to be beneficial. And so we haven't actually done, I wouldn't say any sort of optimization on different levels of expression here. But as we start to add in, so the strain is pretty heavily engineered. There's probably 15 heterologous genes in there at this point. But as we start thinking about adding polypropylene or PVC or all these other things into the same chassis, that will absolutely become a problem that we'll need to look at. So great question.
So, I see another question from Daniel Acosta. Great job. I have question about TP metabolism and transport. Did you look into using either of Pseudomonas strain GO16, 19? This person know about the thing. Known to natively metabolize TPA, and if so, how did this compare to the heterologous system you ultimately employed?
That's another really good question. So
yes.
At the time we were doing this work, the GO16 strain hadn't been published yet, so we weren't aware of that. And I'd love to plug in those genes into our system and compare them head to head. So we don't know the answer to your question of other pseudomonads. We did compare to Rhodococcus and Decomomonas, which again isn't quite apples to apples because those strains are inherently have different growth rates and things. But we found that our strain, TDM461, so the strain that had just the TPA engineering, performed the best compared to the hosts that we pulled from. But it would be really interesting to look at that strain versus GO16.
Fantastic. Actually, I love, because you used to use, the only Pseudomonas for many years, but I'm only using the Rhodococcus, but now two strains now married together. So that's a wonderful kind of observation I saw. And another, I want to ask one quick question because before closing. So from the environmental perspective, your approach to making the product, you mentioned, a couple of products is fantastic. From the economic perspective, the complex process you just suggest and that you're using would cost a lot. The final product actually very cheap compared to the entire process. That's my guess. I already done some preliminary economic analysis for my process. So could you kind of comment on this point? Probably this is a challenging point for everyone working on this field, but I just want to hear from your perspective.
Yeah, so the process models, as opposed to in the lignin space, that there are really well-established process models that, at least at NREL, we're still working out what that process would look like. And so an effort to do techno-economic and life cycle assessment on that kind of pipeline that I showed for the mixed plastics case is underway. And so it's a really good question of what makes sense, like what makes sense from a process standpoint and a product standpoint. And for sure, we'll need to make something of really high value to make it worth it. And I think that's really the incentivization of plastics upcycling is really, I guess I didn't define plastics upcycling ever. We think about it as making a product that's of more value than the feedstock to incentivize the reclamation. So if you think about it, not necessarily, I guess I could speculate, but that's not really worth it. We are currently looking into what would make sense economically and environmentally. So conservation of carbon is a really important thing to think about as well.
Sure. I mean, let's continue to discuss more. Okay, so that's wonderful. So because of time, I want to just close now. So thank you all for joining and staying today again. Although many people are still enjoying extended, long-awaited vacations or catching up accumulated emails like me over the break, we will meet again next week on January 13th, Thursday, the same time. And we will interestingly having another Nature journal editor, Dr. Ross Croney, the Nature Communication Senior Editor, and Dr. Alex Pedorek from University College London, now European person. And as usual, the follow-up Zoom chat will occur via the link in the chat box I just posted. So click that one if you want to talk to Terry or Ellie, and we will discuss more. And then see you soon, and then see you next week again. Thank you so much, everyone.
Thank you.
Thank you.
So if you go to chat, you will see the link over there. And then, as I said, you just click that one, and then basically say yes. I mean, you will go to another room, basically.
Got it.
Okay, thank you, Tony.
Thank you, Dr. Moon.
Okay, Happy New Year.
Same to you.
Hopefully see you, somewhere in person.
Yes, one day.
Yeah, one day. Yeah, because of now, school kind of also kind of limiting, in-person class and other things. So, but hopefully sometime later.
Yes, perfect. Thank you so much.
Thank you so much.
Bye-bye.